Hello everyone, good afternoon. My name is Dave Stevens. I'm Vice Dean in the Faculty of Science and Professor in the Department of Mathematics and Statistics. On behalf of the faculty, I'd like to welcome you to the final Sun and Science session of the summer. So this is an opportunity for researchers and students to present their work. And it's part of the Soup and Science series that we run in fall and winter in normal times. So today we've got four great speakers who will be presenting their research and some outreach activity that's also been going on. So we've got Professor Nicole Li-Jessen from the School of Communication Sciences and Disorders, Imane Chafi who's working in the Faculty of Engineering, Jessica Ford, who's working with the Redpath Museum, and then Professor Judith Mandl from the Department of Physiology. So the presenters will talk for around 5 minutes, and then when we've completed the four presentations, there'll be a question and answer session in which you'll be able to ask questions of the speakers, and Preeti will talk a bit more about that when we finish the presentations. So we'll begin with then Nicole's presentation. So this is Professor Nicole Li-Jessen from the School of Communication Sciences and Disorders. Thanks, Nicole.
Okay, can you hear me? Okay, good. Hello, everyone. I'm Nicole, and thank you for being here today. So I'm going to talk about our work concerning voice and upper airway health and diseases. So what you see here is your larynx, which is a very important organ for your daily communication, swallowing, and breathing functions. So in our lab, we have four major research themes. Because of the time interest, I will only talk about two of them: digital healthcare and tissue engineering. So anyway, in all of these four themes, we do both basic science research, meaning that we work with cells and at the bench. We also do clinical research, meaning that we work with clinicians and patients in the hospitals as well. So if you put your hand here around your Adam's apple, if you speak or say an R, you will feel some vibration on the neck surface. And that is actually the wave or the vibration coming from this very small, tiny tissue we call vocal folds. So what you see here is actually a very slow motion of the vocal folds when you speak. So in general, actually, they are vibrate at a very high frequency, over 100 times per second. And if you speak higher or sing at a higher note, it can go all the way up to 1,000 hertz. So you can imagine when someone who, like teachers or singers, salespeople who use their voices a lot at work, they are very at risk of vocal injury. So I'm going to show you a video for someone has the vocal injury. If I can get the video work. Okay, so I couldn't see the play button here, but okay, here. Thank you, Preeti. And so what you see here, the left side, that is someone with the vocal injury. You can see here someone with a very hoarse voice, and they can run out of their speech very easily. So what we are trying to do here is to avoid or prevent this situation happen. So what we are trying to do is we are going to develop a device, which is a wearable devices, which is basically a sensors that putting on the neck surface. And that is a connection to your smartphone, which is an app we are trying to develop. The app is based on the machine learning algorithm, which is to detect whether someone and predict someone which is at high risk of vocal distresses. We are also trying to develop another algorithm to detect other upper airway distresses, like coughing, because coughing is a very common symptom for people who have asthma, sleep apnea as well. So we would like to develop this algorithm and devices to monitor this situation and conditions and tell the patients and the physicians that someone is at risk of certain distresses and upper airway diseases. The other project we are working on is tissue engineering. What you see here is a cancer growth on the focal folds. So for patients develop a cancers on their larynx, usually their chemo drugs and radiotherapy has to dial down a lot because it's so close to our brainstem, and we are trying to avoid any side effects to the brainstem, which is a very vital organ of our human body. So what we are trying to do is to develop a biomaterial which can carry the anti-cancer drug like chemo drug or any immunotherapy drug that we can directly inject into the focal folds here to localize the drug effect only to the focal folds and prevent any possible negative and side effects to other human parts of the body. Once I'm getting into this idea, and then I realized that it's such a complex problem to solve. There is a lot too many cell interaction with the biomaterial design, and that we have to test at the bench is going to be impossible. So I decided to do it using the computer to do part of the work. So the idea is to develop the computer model to predict which biomaterial design can carry the best anti-cancer drug effects. And so we are developing such a computer model to simulate the anti-cancer drug effect specifically for the focal fold cancer. So again, we have a lot of projects going on in our lab, and I hope I can have another opportunity to talk about this or other exciting projects with you next time. So thank you.
Thank you very much, Nicole. So our next speaker then is Imane Chafi, who's been working on a project in the Faculty of Engineering.
Yeah, so hi everyone, my name is Imane Chafi, and today I'm going to present to you my research as an undergraduate student with the Faculty of Engineering. So I'm going to start with resuming the slideshow and tell you first, I have made a lot of research in computer science, but also in software engineering. And the first one I've done is a software web interface to classify musical neumes. So the first thing you might ask yourself is, okay, but what is a musical neume? Actually, a musical neume is a very ancient, so with the medieval times, it's a note, a musical note, that is actually square instead of being round. And what's happening right now is that musicologists around the world actually want to have access to musical notes and musical manuscripts. However, the manuscripts are all in square notation that can be processed with a computer. So what we were trying to do here is actually to give the musicalist actually an interface to see the musical notes as encoded inscriptions. So what we did is we asked musicologists if we could have pictures of the manuscripts, the original manuscripts. And we were able to scan the manuscripts and see where the notes were depending on the staff. So neumes are actually made in a very different fashion than notes. They're actually very close to one another. So for example, if you have two notes that are doubles, they would actually be very close to each other, touching. So what we would first need to do is to actually separate the notes from the staff from the actual image. And then afterwards, separate the notes that are touching each other to know which ones are which on the staff, and also to classify them afterwards with this web interface. So what I was able to do here is not only to provide musicologists with a way to see the neumes as codes, and afterwards they could import it, download it, and put it inside of a software as a MIDI file. MIDI files are in music files where you could actually put it in MuseScore if you know the software to actually listen to the music. So now they could listen to manuscripts from medieval times without having to go through calling the musicologist in Austria, asking for a musicologist to transcribe everything by hand. So that was really helpful because everything is automated here. And one of the features that was actually really funny with this is you could also edit the Neumes. So for example, if you import all the musical Neumes from manuscript inside of the software, and then it gives you all of the code, and you see you're a musicologist, you enter, the website is actually live if you guys want to go see. It's cress.staging.simsa.com. And you can actually see all the Neumes and see all the codes that the software did. And if you're a musicologist, you go in and you see that there's actually an error in the code. You can go and fix that. So if you can see here, this is where the code would be. And if you see that there's a mistake, you can flag it. And you can also ask, put comments, ask people working in the software, because that was last summer, my project from last summer, so I ended it. But now there's more people working on it, so if you see that there's an error, you can tell them, and they'll be able to fix it. So that was my project. I was really happy that I was able to complete it in the summer, because most of the time it's very difficult to finish something. And it's still going. So if you guys are interested in musicology, it's a very niche system. So there's not that many people inside, but it's really, really interesting. And that was with the SIMSA labs. You can check them online too. They do a lot of cool things too. If you're more towards, so that was more software, but if you're more towards computer or electrical engineering, what they're trying to do is create a musical instrument without there being a musical instrument. It's basically just a machine, and the way you touch it, so it's more haptic wearables. So the way you touch it would make different sounds without having to put voice, without having to put breath in. So it's really a musical instrument that's all computerized. That's very interesting. So I had also a video. It's not working right now, but you can see this was sort of the interface. It's also online if you guys are interested. And finally, right now, I'm working on COVID-19 research, and I'm working with the Li Lab. We're a computational biology and machine learning group. And what we do is we analyze data. So all the data that's in GFIN. So GFIN is the global Canadian data resource for health. And then there's also WHO. So the World Health Organization, we get all of the data from COVID. And what we're trying to do right now is to find exactly when did the public realize that COVID was happening, and exactly when did official sources from different countries realize that COVID was actually beginning as a pandemic. So we're looking through all of the official statements that are coming in through WHO, GFIN, and also CoronaNet. That's another database. And we're trying to find topics. So it's very much a lot of epistemology. So we're looking at words, the relation between words. So for example, if a person was writing in Twitter, oh, this person had pneumonia, and then we get that article, then we can see, okay, pneumonia, we can see where the Twitter was actually made, and then we can make a link with that with other articles that have been sent to see, was it actually starting then, and to actually see at which point in which countries there was an increase in understanding that COVID was beginning to become a pandemic. So that's all for my research. Thank you for listening, and I'll go to the next presenter. Thank you.
Thank you very much, Imane. Our next speaker then is Jessica Ford, who's going to talk about some outreach work done through the Redpath Museum.
I cannot seem to get the screen to work. Sorry about that. There we go. Okay, great. There we go. Okay, cool. Sorry about that, guys. So hi, I'm Jess. I'm a PhD student in the Redpath Museum studying biology. And I'm actually not telling you about my research today, but about a diversity initiative that I'm involved in and that you could get involved in too, if you like. And it's called STEM Diversity at McGill. So STEM Diversity at McGill is a student-run initiative and exhibit that's housed in the Redpath Museum. And it promotes diversity and inclusivity in science, technology, engineering, math, and medicine by highlighting the achievements of underrepresented groups in these fields and by letting them tell their stories. Our exhibit exists in two parts. We have an online exhibit and a coloring and activity book for kids. So our online exhibit is accessible to anyone with internet access and is usually also housed on touchscreens in the Redpath Museum, but unfortunately the building is closed right now. And it features interviews from women and underrepresented groups in STEM fields where they can tell their stories, as well as resources about some of the barriers and injustices in STEM fields, and links to other groups that we want to promote. And you can access it here at this little link whenever you want. We also have a coloring and activity book that I authored and illustrated, and it features underrepresented groups in STEM as well, and activities related to their research, as well as some fun coloring activities like draw yourself as a scientist style activities. These books have been distributed to schools, libraries, special events and programs, all for free, thanks to the donations from our donors. And we've distributed over 1,200 copies so far. So they've been really well received and have been going a long way. But we don't want to stop there. We have some big future plans coming up, and we are looking for help and some new ideas with these. So we have some upcoming plans, such as a new website we are making that has more information about us and about the barriers that currently exist in STEM fields, as well as some new features. We are also making a lesson plan to accompany the coloring book when it goes along to schools, and are planning for future outreach with schools done by the members of STEM diversity at McGill. And the point of our outreach is not to make the students we work with understand how chemistry works exactly or anything like that. It is simply to give them positive experiences with science and with scientists, and show that science is something that can be accessible and understandable to them, and isn't just something done by old white men in lab coats. So if you want to be involved, we would love to have you. You don't have to be in a STEM field. You can be a graduate student or undergraduate. It doesn't matter. We welcome everyone. You don't need any prior experience. So if you want to get involved, you can reach out to me through email or Twitter, or to my co-chair, Charles. I'm always happy to hear from you.
Thank you very much, Jessica. The final speaker today then is Professor Judith Mandl from the Department of Physiology.
All right, hopefully you can see my screen. So I'm a cellular immunologist, and most of what my lab does is study immune cell migration, but one of the projects that we've been working on that I think is particularly pertinent right now is that we have an ongoing project looking at immune responses in bats. All right. So if you've been following the COVID-19 news, then you will probably know that the closest identified relative of SARS-CoV-2 was found in bats. The particular strain that shares about 96% sequence identity is RATG13. That was sequenced from a rhinolobus bat in 2013 in Yunnan province. And there are other coronaviruses that have been sequenced from bats that also share similarity, but this is the most similar one. And even though it's the most similar one, incidentally, just to point out, is still is about 20 to 50 years in terms of evolutionary time to the current SARS-CoV-2 that is causing the pandemic. Interestingly, bats are reservoir hosts for other viruses as well, some of them very high profile ones because they're very pathogenic, such as Hendra and Nipah viruses that circulate in Malaysia, Northern Australia, and other Asian countries. Of course, Ebola and Marburg viruses that we heard a couple of years ago in a big outbreak in Africa. And I'm sure you know that bats can harbor rabies virus even here in Canada. And even in addition to these very high profile viruses, it's thought that bats are the origin of, or harbor the origin viruses of a number of human viruses with which we have more longer term evolutionary relationships, such as measles, mumps, hepatitis B and hepatitis C. And what makes bats so fascinating is that none of these viruses cause disease when they infect a bat, as far as we know. And in addition, all of these viruses share a common theme, which is that they encode their genetic material using RNA rather than DNA. So what my lab is interested in is asking, are bats special? So bats are special mammals in a number of different ways. They are the only mammal group that flies. They echolocate, they hibernate. They have exceptionally long lifespans given their very rapid metabolism. They live in enormous colonies in some instances. But what we really want to understand is whether they're special from an immunological perspective as well. So what makes them so resilient to disease when they're infected with RNA viruses? And we think that we can learn something from that that might help us treat people that get sick with viruses that jump from other species for which we might not have vaccines available, and as you are probably aware, take quite a long time to develop. So back in 2017, I sent a very brave PhD student to southern China in our first pilot field immunology study. And what she did is she captured wild Hippocytrus armiger bats, shown here. This is Caitlin giving a bat that she captured some water. And then we bled these bats from the artery in the wing to take some blood. And we were probing cells in the blood, the white blood cells, to understand how the immune cells in bats are responding to specific stimuli, that we have a comparison with humans and mice that we know very well how they respond. And so far what we found is that bats are really quite different in terms of how their immune responses progress. And this is something that we're following up in my lab. So this is currently what my lab looks like. And just to point out Caitlin in particular, this is her in her full PPE as she was going into the bat caves. Of course, I did not want her to bring back a new virus from southern China. And these are our funding sources and our collaborators that made that study possible. Thanks.